Thanks for the invitation to speak today. So today I will be talking about nanotechnology and microfluidics to develop targeted therapies. And first of all, I'm an assistant professor at the College of Pharmacy at University of Manitoba here in Canada. And that's what my lab is doing. So we work at the intersection between nanotechnology and microfluidics. And the goal of my talk today is not really to introduce nanomedicine to you. Hopefully you have an idea about that, but to really convince you about two aspects. First is protein corona. Second is the effect of shear forces or flow conditions or the dynamic environment, and how we should consider both of these two factors in evaluating new therapies in order to improve the clinical translation of new drug delivery systems. Okay, so what we do in the lab is similar to a typical nanoscience or nanomedicine lab. So we design lots of nanoparticles of different dimensions, different shape, different surface porosity, chemistry. For drug delivery applications, we are always interested in encapsulating or loading them with different drugs and determining their properties and release kinetics. So we do lots of studies on that. We also are very interested in how these nanoparticles are able to cross biological barriers, which is one of the main advantages of using nanoparticles as a drug delivery system. Not only barriers, but also how they get transported from one environment to another within our body. So how they transverse from the blood circulation, for instance, to the extravascular environments until they reach their target cells. And we're also interested in biocompatibility or toxicity screening, because as the case for development of any formulation, you want to make sure that it's not going to kill the patient and it's going to be safe. So we do lots of studies on how the nanoparticles interact with the different intercellular compartments. And we also introduce some simulation models in order to develop a clear idea about the safety of these nanoparticles and be able to extrapolate the results that we get in the lab into more authentic context. And finally, we are interested in efficacy, like whether the drug delivery system would be successful or not. And these kind of assessments really depend on the application of the specific delivery system that we develop. This is an example, for instance, of a product that we have developed to target intracellular compartments using peptides that mimic the bacterial invasion proteins. And actually, this project was one of the most successful stories that we had, and it's now being translated by a company in Germany. Okay, so what we are focusing on in my lab right now is really digging into the biological identity of nanoparticles. But what is this biological identity? So just in brief, when you prepare nanoparticles, or when we prepare nanoparticles in the lab, they come with a set of properties. We sometimes call them the engineered properties of nanoparticles. But now when you inject those particles into the blood circulation, they develop new set of properties because those particles interact with the surrounding environment, with the proteins, with whatever components in this biological environment. And this results into a new identity. So we call it the biological identity of nanoparticles. And now these newly developed identity dictates the behavior of the nanoparticles. So in order to develop nanoparticles that have high translational power, you need to really dig into this relationship between the engineered properties and the biological properties in order to be able to predict their behavior precisely. That's one aspect. The second aspect is that our body is dynamic. It's not really static. So you need to really investigate or do your evaluation under conditions that is dynamic. So you're very interested in the trafficking of the nanoparticles. And that actually brought us to the microfluidic part where we develop microfluidic chips to really simulate these dynamic environments and also simulate the microstructure of the biological barriers in the lab using those small chips that we will talk about today. And we have different applications, so it's not only cancer targeting as it's the case for most applications of nanotechnology, but we are also interested in developing therapies that are safe to use during pregnancy. Okay, so let's start talking about protein corona, which is the first topic here. So here is a nanoparticle that we inject in the circulation. Those nanoparticles interact with the mainly different components in the plasma, including the plasma proteins. So mainly there is a corona deposited on the surface of those nanoparticles. It's not really a covalent linkage, it's really a dynamic system as well, an equilibrium where the protein forms a corona in its equilibrium system, and we call this protein corona. And now we know by like our work as well as by others that this protein corona dictates nanoparticle behavior. What we don't know is that how about so what is exactly known is that when nanoparticles interact with the plasma proteins, they come up with a different set of properties that they affect their fate, their biodistribution, etc. But not really evident yet or understood yet how the nanoparticles interact with the other components of the system once they leave the blood circulation. And this is what we started to focus on recently, is how nanoparticles interact with other extravascular proteins like the collagen, which is the main protein that is found like in the body, the main protein. So we did several studies using different nanoparticles with different properties, and I will share today the results that we recently got using gold nanoparticles of different shapes and sizes. So we characterized them and we prepared them and characterized them. And the reason why we use gold nanoparticles here, because they come with specific optical properties that allow us to really investigate their change in surface properties. And these properties are called surface plasmon resonance or localized surface plasmon resonance. And this is the peak that we get simply using a UV spectroscopy. So any change in this peak, whether it's broadening or it's redshift in the peak, this really indicates that something is happening at the surface of these nanoparticles. So we did our studies with the protein, and we do lots of measurements to determine binding constants. Today, I will only show the optical measurements. So simply, if we have small particles and large particles, let's say in this study, it was like 15 and 30 nanometers, and then we expose them to a protein, we found like the first thing that we observed is that there is a redshift. And this redshift increases with the concentration of the protein. So there is clearly a protein corona formed on the surface of those nanoparticles. But what we also observed is that the large particles were behaving differently than the small particles. And it's not just different, but even the response is very different. So for the large particles, we found that at high concentration, the shift was not as much, but it was like similar to what we observed in small gold nanoparticles. But at the lower concentration, we found some broadening, which indicates the aggregation of the particles. And this was a little bit weird here, but we're doing some experiments, but we have a hypothesis right now that explains what is happening, is that for the large particles at high concentration of proteins, they form really nice layer of proteins around them that really keep them dispersed. But at low protein concentration, they could form some bridges between those nanoparticles that could really make them or induce some kind of aggregation, but we're still not sure whether this is the case or not. But overall, we see nanoparticle dependent behavior. We also see a concentration of protein dependent behavior for these kind of interactions. But then we were interested to see whether we will see different protein corona for the different proteins. And the naive assumption was that, all proteins are formed of amino acids, really in small changes. Maybe they all interact similarly, but we didn't see the same. we see very different behavior. And without going into lots of details here, so like, but we tried several concentration from the light color to the darker color here. And as you can see, I mean, the shift is different. So here it was more of about 5 nanometer shift, and here it was like 10 nanometer shift, and the pattern is also different. So clearly, So clearly there is a difference, and we've done set of experiments based on fluorescence as well to determine the binding constant, and we could clearly see that the collagen is binding more efficiently to the surface of gold nanoparticles more than the albumin, which is the main protein found in the blood circulation. But then the next logical question is that if you inject nanoparticles in the blood circulation, and then they move from the circulation to the outside, to the extravascular part. Let's say if those particles are targeting a tumor, and then the nanoparticles will exit the circulation to the tumor tissue. So will the albumin coat or the plasma protein coat be exchanged by collagen or not? And this is very important because we want to see what is the identity of the particles that will interact with the cancer cells. And based on that, we will be able to develop therapies that have high interaction tendency with the cells. So we did the same experiments. We exposed the nanoparticles to albumin first, and we got a shift that is similar to the shift that we observed with albumin. So that's just a normal experiment. And then we added collagen on top of that and see whether we will see a change or we will not see any change. This means that the collagen did not replace the albumin. And actually we saw a change that is really relevant to when the nanoparticles interact with the collagen. And doing several experiments, we could really prove that collagen actually displaces albumin, and it's really a dynamic protein corona. And we shouldn't really think about it as the first protein that interacts with the particles will always stay there and will dictate the behavior no matter where the nanoparticles are. And then we even thought about a different scenario. So let's think about a different application rule. So if we inject the nanoparticles not in the circulation, it's not like intravenous administration, but how about we do intradermal? So we inject the nanoparticles, for instance, in the skin, where we have lots of collagen, or in any other site. So it's mainly in the extravascular first. So then the sequence is different. So they are exposed maybe first to collagen and then to plasma proteins here, albumin. And I just want to note here that it's really not really understood how this happens in the reality. So we are doing lots of simplifications in these studies in order to really understand the mechanistic story before we go for complicated systems. But nevertheless, we did this experiment. So we started with collagen first, and we got a shift that is equivalent to the collagen. And then we exposed this to albumin, but we didn't see any change. It was overlapping curves, as you can see here, and it was really the same as collagen-treated nanoparticles. So the binding affinity of collagen is way more than albumin. And we need to really start thinking about protein corona as not just considering them in the study as like simply, but really thinking about the whole journey of the nanoparticles and try to imply this in how we evaluate our nanoparticles. Okay, so hopefully I could convince you how important is protein corona for evaluation. The second is it's not just protein corona, but it's the whole environment, and the whole environment is dynamic. So when the nanoparticles are injected in the circulation, they interact with the cells not under dynamic static conditions, but under dynamic conditions. And this affects how the nanoparticles really interact with the cells. So going into the biophysics of how the nanoparticles drift from the circulation and how they come in contact with the cells, but also it is also well known that shear forces, or the force that the flow creates on the cells, would really affect the cell phenotype. So there could be downregulation or upregulation of specific receptors that affect how the cells interact with the nanoparticles, but also affect how the nanoparticles impact the cells either positively or negatively. So we did some studies to really understand how the nanoparticles interact with the cells under different conditions, different shear conditions, like shear stress conditions, as well under different types of flow, whether they are laminar, so they are parallel and very, this is the normal physiological conditions, or under disturbed conditions where there is a pathological reason behind that. And so we used a model called zebrafish model. It's a transparent model in this case. So we were able to do some confocal studies and really image the larva and see how the nanoparticles flow. So we did several experiments, and using some flow dynamics simulation, we could really analyze the shear forces at different locations inside the vessels, and really translate this into quantitative number. And what we could conclude from the study is that we found the highest accumulation of the nanoparticles in area of branching, where there is some disturbance in the flow and sometimes circulating flow, as well as when they're within a specific range of shear forces. But at this level, we thought that we have lots of restrictions. First of all, I mean, we're not really simulating the human completely. It's a very different model than the angiogenic blood vessels in tumor targeting. And also, we are very bound in terms of what we get, right? It's not like we are able to design experiments based on a set of parameters so that we have these kind of understanding of how nanoparticles interact with the different cells under different conditions. So, and this is actually what moved us to use organ-on-a-chip models to really simulate, well, to first of all use human cells and really create this microstructure inside the chips, and also to be able to precisely control the conditions that mimic the physiological conditions. So what are organ-on-a-chips? These are microfluidic chips, as you can see here. And these microfluidic chips have microscale channels of different designs, different sizes within the microscale for sure. The idea here is that you are, as if you are taking a microscale section of the biological barrier and trying to replicate it on a slide, on a chip. And so you are mimicking the microstructure of the human biological barrier exactly. And you are exposing this to a flow of fluids that is going through a microscale channels. And if you think about our microcellular structures, these are microscale channels as well. So we are mimicking what is happening inside our body, and we're exposing those cells and tissues to flow conditions at the same level and same behavior of as in real case conditions. So this is the system that we have in the lab. So in the middle here, there is this is a running experiment of a chip connected to flow of fluids, and we have here 2 channels, and you can see there is inlet and outlet for each channel. And what we can do in the lab is that we can actually run several circuits independently of each other. Each of those circuits would have its own flow rates and flow conditions. And it's as you can see, it's a very complicated system, but we can really, using a computer or a software, we can really have a code that really control these experiments for a long time in order to conduct long-term experiments. And this is a closed system, which is similar to our body. It's not like an a start and an end. It's really a closed loop, similar to what we are seeing in the circulation in our body. And using a flow controller and sensors, we're able to really accurately control even very slow or very low flow rates. But for these experiments, the step number 1 is definitely the chip fabrication itself. So we use a polymer called PDMS. This polymer is really flexible and very transparent, so it allows us to really do good imaging. And we use, without going into the details of the fabrication, but it's based mainly on lithography. So we develop a mold first, and then using this mold, we use it to develop those two parts of the chips. And then using we use a polycarbonate membrane at the end, and then we bond them together using plasma cleaning that we have in the lab to develop those microchips at the end. And this is like real pictures of some molds that we have in the lab of different dimensions. And as you can see here, these are different chips of different designs. And this is the one batch of chips that we have developed here. And this is when we connected the chip to the circuit. Okay, so I will give like two brief examples of how we are using those microchips. The first is dynamic vessel on a chip model to see how the nanoparticles interact with the endothelium, which is the first barrier that the nanoparticles get exposed to when injected into the circulation. So our main idea from this project is that we want to develop a diagnostic nano tool in order that is really safe to use. And one of those probes that are commonly used or known is the quantum dots. They are very bright particles, but the problem is that most of them are based on cadmium cores that are very toxic. And our idea is to, or I mean, based on other work as well, is to use indium phosphide quantum dots that have been proposed and used, but the toxicity on the different cells is not really well studied so far. So that's what we did. So we developed those cores and we coated them with polymer to enable their transition from the organic solvent to the aqueous phase so that they are able to be used for biomedical application. And we were successful to do that. And we were able to design different formulations that they were all fluorescent and they were all stable for a long time. But then after preparing the system, we then moved to our vessel on a chip model. And as you can see, it started with very simple model where we have the endothelial cells grown in one channel. And then those channels are connected to the cell culture media and they were flowing. And then we exposed this to the nanoparticles. So we exposed the endothelial cells to nanoparticles, and then we did a series of studies to understand the response of the nanoparticles to those nanoparticles. And by no means, I mean, we were expecting this already, that in case of cadmium selenide quantum dots, which is the conventional type of quantum dots, they were toxic, especially going to higher concentration. But for the indium ones, they were all safe and there were no significant difference in terms of cell viability compared to the ones not exposed to nanoparticles at all. And we did a series of studies that we're still working on right now. And so we did a transcriptomic study to even go to the genetic level and understand how the cells respond. And we saw differences, but what was really interesting to see is that well, cells really behave differently when they are in dynamic conditions versus static conditions. And they behave differently in response to nanoparticles under dynamic conditions versus under static conditions. So it's really important to really consider the shear stress effect when we are evaluating nanoparticles and include this in our evaluation platform. The second example is placenta on a chip model. And this is why we were we are interested in developing safe therapies during a pregnancy. And the challenge here is that this population, unfortunately, with the pregnant women is not really studied well. So most of the novel therapies are not really developed for this population because it's difficult to really include pregnant women in studies. So it was very important to really develop a model that really mimics the human scenario so that you're able to develop these therapies and be able to really translate them later on into clinical trials based on really strong data that is relevant to human pregnant women. So that was the case. So we developed a system that was more complicated here and based on our pressure system. And we had maternal cells, placental cells on one side, and fetal endothelial cells on the other side. And we developed the model, and we actually found not just small changes, but really big changes between the static model and the dynamic model. So as you can see here, there is kind of like lines here that really models or represents the microvilli, which are protrusions that are known to form on the epithelial cells under shear stress, but under static conditions, they were not really formed as much. So really the dynamic conditions really mimic the human placenta way more than using static models. But not only this, we also did a set of experiments to see the barrier function, and we could see that under dynamic conditions, a minimal concentration of the fluorophore, which is fluorescein sodium, could really cross the barrier here. So this is a better barrier than the static model. So the barrier function is much better. So we are doing lots of projects now using this model. I'm not going to go into details, but in one of the projects that we are doing, we're trying to develop microRNA loaded lipid nanoparticles for targeting a fetal anomaly. This is a project that we are working on right now. And I just want to mention here that there is another importance from the use of microfluidics, because microRNA, as you might know, is a really unstable molecule. So you need to really encapsulate them in a condition that is not harsh. So you can't really use organic solvents or batch methods or homogenization to really prepare these nanoparticles. And that's why we're using another setup that we have developed in the lab, also based on microfluidics, and several labs are using it already, where we are developing purging the systems into very small channels of different designs that lead also to the formation of nanoparticles. And this is a video of a running lab here when the two liquids, the aqueous phase and the organic phase are, or the ethanolic solution, they are moving here in parallel under laminar conditions, and the nanoparticles are formed in between the two layers here. So it's a very short presentation, but hopefully I could really convince you that protein corona and shear stress are really important to consider. And organic and organ-on-chip models really mimic physiological conditions. I would like to thank my team. It's not, of course, it's not me who did all these experiments, but they all did, and I'm just reporting them. And I want to thank the funding agencies, and also highlight that there are some open positions in my lab. Thank you.